Eukaryotic transcription begins when RNA polymerase II binds to the promoter region of a gene. A crucial part of this initiation process is the recognition and binding of the TATA sequence, a short stretch of DNA rich in thymine and adenine nucleotides. The subunit of RNA polymerase II that binds to the TATA sequence is called the TATA-Binding protein. The TATA-Binding protein binds to DNA using an 8-stranded beta sheet that rests atop the DNA helix like a saddle. 2 protein loops drape down the sides of the DNA like stirrups. Binding of the TATA-Binding protein introduces a severe kink in the DNA backbone. This kink dramatically bends the DNA helix by nearly 90 degrees and is thought to provide a signal to assemble the rest of the transcription complex at the initiation site.